Hi, my name is Dr. Dominique Cagle, and I'm a technical information scientist here at the Jackson Laboratory. I'd like to thank you for joining us for our presentation today, where we will be discussing using inbred mouse models for infectious disease research. Before I begin our presentation today, I'd like to quickly outline what it is we'll be discussing. First, we'll go through the benefits and limitations of using inbred mouse models for infectious disease research. Recently, there's been a lot of attention and focus on using humanized mouse models for infectious disease research. There's a lot of utility in using these models. However, today I'd like to share with you the utility of using inbred strains for infectious disease research. And I'll highlight a number of examples, including tuberculosis, salmonella, anthrax, and influenza infections being modeled in inbred mice. I'll also share with you how inbred mouse models have been used for vaccine development and where you can go to find more information regarding these mouse models using the mouse phenome database and how you can use this information to better select your strains. Finally, I'll share with you the importance of using genetically stabilized mice for your research. First, let's go through the benefits and limitations of using inbred mouse models for infectious disease research. There are several characteristics of inbred strains that make them good models for studying infectious diseases, including the fact that they are susceptible to many of the same diseases that infect humans under the right experimental conditions. Inbred strains also display a range of susceptibilities to different pathogens, and this reflects the genetic diversity seen in humans. You can take advantage of these differences to study the different genetic factors that contribute to disease and host response. Inbred models are also amenable to genetic studies. It's relatively simple to make genetic mutants and thereby determine their contribution to infection. Also, inbred mice have fully intact immune systems as compared to their immunodeficient mouse counterparts that are reconstituted with human immune systems. Inbred mice have immune systems that are within their own natural environment, and we'll see an example of how this can play a role in disease outcome. The availability, cost, and scalability of performing experiments with inbred mice is unsurpassed, as inbreds are some of the most inexpensive models to use. And finally, inbred mice minimize genetic variability. Because mice from one particular genetic background are genetically identical, they should respond to treatments uniformly. And so you could take advantage of these differences in different inbred strains by creating cohorts of mice and testing them with your different compounds. So because mice from one particular genetic background are genetically identical, they should respond to treatments uniformly. And when you pair this up with mice from different genetic backgrounds, they can be used to study the genetic differences that contribute to disease. An additional advantage to using inbred mice is that many of them are a part of our genetic stability program, and this ensures that the mice that you receive from us are not only of the highest genetic stability, but you're ensuring the reproducibility of your results when you use these strains. So some of the limitations of using inbred strains for infectious disease modeling is that some pathogens cannot infect mice, and that's where humanized models have a clear advantage. Also, some pathology seen in mice may not reflect the human disease, and along those lines, there may be differences between mouse and human immune responses. So you have to take all of this information into account when you use these models. And finally, some risk factors for infection cannot be modeled in inbred mice, and I'll show you some examples of this later on in our discussion. So as I mentioned, inbred mice have their own immune systems in their natural environment, and this can impact disease progression in some models. In this example, BALB/c mice, which are an inbred strain, were infected with a hepatotrophic virus tagged with luciferase, and they were compared to NSG mice that expressed a mouse immune system, NSG mice that expressed a human immune system, or NSG mice that expressed a human immune system that were also HLA restricted, and this increases T cell response. And so all of these strains were infected with this hepatotrophic virus that was tagged with luciferase, and luciferase expression was measured as an indicator for viral replication. And we can see that BALB/c mice were able to clear infection by day 10, and NSG mice that expressed a mouse immune system were able to clear infection by day 20. However, NSG mice that expressed a humanized immune system failed to clear infection by day 20, indicating that although these humanized strains, are great tools for studying pathogens that typically only infect humans and have been difficult to study in other models, they would benefit from further optimization, and those studies and experiments are currently underway. So now that I've told you about the benefits and limitations of using inbred strains for infectious disease research, let's go through some examples of some pathogens that have been modeled in inbred mice. First, we'll start with tuberculosis. Tuberculosis is a global health concern, and it's actually the second leading cause of death worldwide, with the majority of cases being in sub-Saharan Africa and Southeast Asia. So there is no vaccine for tuberculosis, and multi-drug resistant strains are becoming more and more common. So it's important for us to gain a better understanding and develop new treatments to this disease. Inbred mice can be infected with tuberculosis via inhalational or intravenous administration of lab-adapted strains, including the highly virulent Erdmann strain and the H37RV strain, which is an attenuated strain, as well as primary isolates. The readouts for tuberculosis infection include measuring survival, as well as bacterial counts and lung histopathology. So as I mentioned previously at the beginning, inbred strains display a wide range of susceptibility to pathogenic infection, and this is true for tuberculosis as well, as we'll see in our other examples later on in our talk. So we can see that C57 Black 6J and BALB/CJ mice are relatively resistant to tuberculosis infection, whereas DBA2Js, CBAJs, C3Hs, 129X1s, and AJs are all sensitive to tuberculosis infection. Here's an example of a comparative study where C57 Black 6J mice and DBA2J mice were infected with different strains of tuberculosis. They were infected with either the H37RV strain or 4 different clinical isolates of tuberculosis, and then bacterial loads were measured in the bronchial-associated lymphoid fluid, the lung, and in the spleen, and we can see that DBA2J mice, as indicated by the white circles, had much higher bacterial levels in each of the tissues examined and for each tuberculosis infection, whether it be the reference strain or one of the 4 clinical isolates. And all of this together indicates that Black 6J mice are better able to control replication of tuberculosis infection in all of the tissues examined. One common feature of tuberculosis infection is the development of granulomas, which are collections of macrophages that block off the spread of infection. And so the authors wanted to determine whether or not these mice can display this clinical symptom. So they looked at lung histopathology, differences between the Black 6J and the DBA2J mice, and you can see that at 9 weeks post-infection, they are able to develop these granulomas. And when we take a closer look at higher magnification of these primary granulomas, that the Black 6J mice, you can see necrotic cell death within these granulomas, whereas the DBA2Js, you see they're much smaller, as well as more neutrophilic content within these primary granulomas. You can also see secondary granuloma development in the Black 6J mice, much more markedly than the DBA2J mice. And you also see tertiary granuloma development with foamy macrophages within these granulomas in the Black 6J mice that you don't see in the DBA2Js. So in another study, Black 6J mice and DBA2J mice were infected with a low dose of the tuberculosis H37RV strain, and bacterial loads were measured in the lung between these 2 strains, and we can see that Black 6J mice had much lower bacterial levels in the lungs as compared to the DBA2Js. And when they looked at gross lung pathology, you can see not only that lungs from the DBA2J mice are much larger than the Black 6J lungs, but they also have much larger and more diffuse lesions in DBA2J lungs as compared to the black 6 lungs. And when they looked at the contents of these lesions, they found that the black 6 lesions contained mostly macrophages, whereas the DBA2J lesions mostly contained neutrophils. They also performed acid-fast staining for bacilli, and they see that DBA2J lungs have much more higher degree of staining as compared to the black 6 lungs, indicating that black 6 J mice are able to better control tuberculosis infection. In another study, black 6 J mice and AJ mice were infected with a moderate dose of the Erdmann strain of tuberculosis, and we can see here that during the course of infection, black 6 J mice were able to survive the infection, whereas 30 days post-infection, all of the AJ mice died. So looking at the bacterial loads, we see that black 6 J mice were much better able to control infection in the lungs, spleen, and in the liver as compared to AJ mice, where they were unable to control infection, and by 30 days we see 100% mortality. In the same study, the authors also infected AJ mice and C57 black 6 J mice with a low dose of the Erdmann strain, which closely represents a chronic model for tuberculosis infection. And so this is commonly seen in the human population, where you see an initial rise in bacterial loads that come down over time, and then, as we can see in the AJ mice, they slowly start to rise again, indicating a chronic infection. In the black 6 J mice, however, you see that these bacterial loads remain relatively constant in the lungs. So this indicates that not only do these strains display the ranges of susceptibility commonly seen in human patients, but they also can be used to model different types of infection. So mice were also infected with the Erdmann strain intravenously, and then a time course analysis of the lung was performed, and we could see that 3 days post-infection, black 6 J mice had much lower levels of immune cell infiltration as compared to the AJ mice. We also see that at day 7, they have black 6 J mice develop these primary granulomas, and even at day 11 we start to see a secondary granuloma forming, whereas in the AJ mice, we see granulomas starting to form, but they never fully form in the way that they do in black 6 J mice. So now that we've seen some examples from tuberculosis infection, let's also look at salmonella. So salmonella is a major health problem in many developing nations, and it causes gastroenteritis, typhoid fever, and non-typhoidal salmonella bacteremia. There's currently no effective vaccine available, so mouse models can be used to better understand this disease and develop new vaccines for it. And actually, towards the end of our talk today, I'll show you an example of salmonella infection and vaccine development using inbred strains. So Salmonella typhimurium can infect mice, inbred mice, if injected intravenously. So while Salmonella enterica cerevar typhimurium is not the causative agent of typhoid fever, it can be used as a general Salmonella infection model. Salmonella typhi, which does cause typhoid fever, does not infect mice, and this is due to the presence of TLR11, which recognizes flagellin and profilin. The readouts for Salmonella infection are survival, measuring bacterial counts in the spleen, liver, and measuring tissue pathology. And as I mentioned, Salmonella infection causes gastroenteritis, and this is one of those disease pathologies that cannot be modeled in mice because this is due to the fact that there are differences in the gut microbiome between mice and humans. Also, the genetic determinants of host susceptibility have been discovered in mice, including the NRAMP gene, which is responsible for restricting the growth of intracellular pathogens, as well as MHC and TLR4. So as we've seen for tuberculosis, inbred strains show a wide range of susceptibility to Salmonella infection, and this depends on the dose administered and how fast they clear the infection, in addition to the genetic determinants that we just talked about. So some experiments were performed to determine the differences in susceptibility to Salmonella infection between 129X1s, MOLF EIJs, and C57 Black 6J mice. MOLF EIJs are a wild-derived strain with very divergent genetics, and they can be used for genetic mapping studies. So in these experiments, mice were injected with a low dose of Salmonella typhimurium intravenously, and bacterial counts were measured in the spleen and the liver. And you can see that in both cases, Black 6J mice were unable to control infection and bacterial replication. We see much higher levels of bacterial counts both in the spleen and in the liver as compared to MOLF EIJs, which are intermediate, and 129 SVJs that were much better at controlling bacterial replication. When they also looked at spleen and liver pathology 4 days later, 4 days after infection, they see much more immune cell infiltration in the Black 6J mice, and we also see necrotic cell death in the spleen of Black 6J mice that you don't see in the MOLF EIJs and the 129 SVJs. So now that we've talked about tuberculosis and Salmonella, let's go through some examples for anthrax infection. So anthrax is caused by the bacterium Bacillus anthracis, and there are 3 different forms of this disease in humans: the cutaneous, gastrointestinal, and inhalational versions of anthrax. And inhalational is the most dangerous because it is fatal and it can be used as a biological warfare agent. So it's really important that we develop small animal models to better understand this disease and come up with a vaccine. So inbred mice can be infected via inhalational infection or intratracheal administration of Bacillus anthracis with the highly virulent AIM strain, and that requires BSL level 3 labs, or the attenuated Stern strain that requires BSL 2 level labs. You can also model anthrax infection in mice by administering anthrax lethal toxin, and I'll show you here in a moment how important it is to know the characteristics of your inbred strain that you're using, because they do show differences in susceptibility even with the route of administration or inoculation. So the readouts for anthrax infection are survival, measuring bacterial counts in the lung, liver, and the spleen, as well as tissue pathology. So like in our previous examples, inbred mice display a range of susceptibility to anthrax infection, not only by inhalation, but also by administration of lethal toxin. So we can see that BALB/CJ mice and C57 Black 6J mice are resistant to inhalational anthrax administration, whereas DBA2Js and AJs are sensitive. However, if you administer lethal toxin, anthrax lethal toxin, Black 6J mice are still resistant. AJs and DBA2Js are also resistant, whereas the BALB/CJ mice are sensitive. And so again, this is really important to know the characteristics and how a particular inbred strain is, the susceptibility of a particular inbred strain to a particular model, because we see, as in the case of BALB/Cs, that are sensitive to lethal toxin administration of anthrax, that they're resistant to inhalational administration of anthrax. So here's another example showing how important it is to use the right strain. In these experiments, the authors measured the survival of different inbred strains after intratracheal administration of the AIM strain. And so although the rate of initial colonization was pretty similar among the strains, you can see that BALB/C mice and B6J mice were the most resistant to infection, and that BALB/CJ females actually lived the longest. And so you can take information like this to develop your own studies, as the authors did here, where they took BALB/CJ mice and infected them with anthrax spores 24 hours before treating them with ciprofloxacin or doxycycline, and then they measured their survival. We can see that the 21-day treatment with ciprofloxacin and the 14-day treatment of doxycycline prolonged survival, and protected these mice from infection. So this is a nice example showing that knowing the differences between susceptibility between the different strains can be useful in designing a protocol for testing the efficacy of different antibacterial drugs during infection. So for our last infectious disease example that we'll discuss today, we'll talk about influenza. So influenza is an RNA virus that causes seasonal epidemics every year, and a significant number of resources are spent yearly on vaccinations and treatment of infected individuals. So obviously there is still a need to develop better therapeutics for this disease. Mice can be intranasally infected with influenza that has been lab adapted, and because influenza doesn't infect mice, the two of the most commonly used lab-adapted strains are PR8 and WSN1, which are both influenza type A H1N1 viruses. Inbred mice are more susceptible than wild mice, and that's due to the presence of or absence of the MX1 gene. MX1 is an interferon-inducible nuclear protein that confers resistance to influenza infection. And so all wild-type mice are MX1 positive, while inbred mice are MX1 negative. However, there are some highly sensitive strains that do not require adaptation, such as DBA2J mice, and they can be directly infected with the 2009 pandemic clinical isolate. The readouts for influenza infection in mice include survival, measuring survival, as well as weight loss. Mice typically lose weight in response to influenza, and so that can be used as a readout for disease progression. The viral titers can also be measured, as well as lung pathology and motor activity. But before I get into a few details of influenza infection modeling in inbred strains, I just would like to mention that a lot of our current understanding of influenza is due in part to utilizing mouse strains to model disease. And so some of the early studies on viral transmission were performed in mice, and many of the most popular and effective antivirals that are currently being used in the clinic today were first shown to be safe and efficacious in mice. And that includes ion channel inhibitors, amantadine and ranitidine, as well as the neuraminidase inhibitors peramivir and zanamivir. So as we've seen in our previous examples, inbred strains show range of susceptibility to influenza virus infection as well. And so when these differences were measured between black 6J mice, BALB/Cs, AJs, and DBA2J mice infected with an H3N2 strain, we can see that all DBA2J mice succumb to infection by day 7 post-infection. When another disease readout, which is body temperature, was measured, the DBA2J mice had the greatest temperature decrease of all the strains, and all the other strains were able to recover from this temperature loss. And finally, when they measured influenza titers in the lungs, we see that all of the three other strains, the C57 black 6J mice, BALB/Cs, and AJ mice, were able to reduce viral titers in the lungs, whereas the DBA2Js were not, indicating that these three strains show much more resistance to infection, whereas the DBA2J mice were much more susceptible. In another study, 7 different inbred strains were inoculated with H1N1 influenza type A virus that was adapted to the mouse, and body weights were measured as a proxy for disease progression. And you can see that in the blue dots that represent AJ mice and the DBA2Js that are indicated by the red dots, you can see that both of these strains succumbed to infection and lost the most body weight, as compared to the other strains that may have lost some weight but then were able to recover their lost body weight. In the same study, the authors looked at differences in susceptibility to H7N7 infection between DBA2Js and C57 black 6J mice. And again, DBA2J mice lost weight and were unable to recover from infection. And we can see that 5 days post-infection, all of the DBA2J mice died due to infection, whereas you can see, as indicated by the green dots, that C57 black 6J mice were able to recover lost body weight and recover from infection. In the same study, they also infected these mice with different doses of H7N7 virus with a low, intermediate, or a high dose, and then they measured survival. And we can see that DBA2J mice died irrespective of the dose administered, as compared to C57 black 6 mice, which at low doses they all survived, but at mid and high doses showed significant decreases in survival. And so this clearly demonstrates that though some strains show levels of resistance, such as the case of C57 black 6Js to H7N7 influenza infection, you can change this susceptibility by increasing the dose administered. These authors also looked at lung pathology, and they saw more severe damage of the bronchial epithelia in DBA2J mice as compared to black 6 mice. And we can see more plugged airways, as well as more tissue necrosis, neutrophil and macrophage infiltration in the DBA2J lungs, whereas in the C57 black 6J mice, we see much more lymphocyte infiltration. And so this is all very important for looking at differences in host immune response to infection. Now let's switch gears and see an example of inbred strains being used for vaccine development. As I've mentioned before, Salmonella causes gastroenteritis, typhoid fever, and bacteremia in young children and immunodeficient individuals, and currently there's no vaccine. So in this study, the authors took serum from 4 susceptible and resistant inbred strains infected with Salmonella typhimurium and tested it against a protein array containing 100 different Salmonella antigens. They also tested serum from infected Malawian children and detected which antibodies were recognizing which antigens on the array, and then they compared these data sets to this infected mouse sera results. Using this approach, they were able to find several candidate proteins that were common between the infected children and in the mice, and so they found that the SSEB protein, or the serodomin secreted effector protein E, of Salmonella was a good vaccine candidate. So they immunized black 6 mice with either no vaccine or an attenuated Salmonella bacteria, LPS to enhance immune response, and LPS with SSEB protein, and then they measured bacterial counts 4 weeks later in the spleen and the liver. And you can see that mice that were immunized with SSEB1 SSEB had much lower bacterial counts as compared to the naive mice, both in the spleen and in the liver. When they also measured survival, you can see that SSEB immunized mice had much prolonged survival as compared to the control immunized and the naive mice. So now that I've shown you how inbred strains can further our understanding of infectious diseases and we can use them for vaccine and therapy trials, I'd like to show you where you can go to find more information about these models so you can make better choices about which strain to use for your experiments using the mouse phenome database. So if you go to our webpage, www.jax.org, and then look on the lower right-hand corner where it says Shortcuts for Researchers, you'll see the link to the mouse phenome database. So if you click on that link, it'll bring you here where you can find lots of various phenotypic data as well as comparative analysis between many of our more popular strains, as well as genotype information for our different inbred strains. But today we're interested in intervention studies. So if you click on that intervention studies link, it'll bring you to this page where you can find a lot of data from different intervention studies that were performed using some of our most popular strains. including their responses to high-fat diets, drug responses, exercise, as well as pathogenic infection. And so you can see here that we have a number of different infectious diseases that were modeled in inbred strains. And so today we'll look at an example from influenza A H5N1 infection. So if you click on that link, it'll bring you to a page where you can find all of the studies done relating to this virus. Some of the information that you can find here include viral loads, as well as weight measurements and other different clinical parameters such as survival, and that's indicated here. We can see in this example that 22 different strains were compared to one another for survival after H5N1 inoculation, and all of these mice were between 6 to 9 weeks of age. And we can see the average survival for each of these strains plotted here. And so this is a really great resource to use when you're deciding which model is more appropriate for the experiments that you are wanting to perform. So I highly recommend that you use this resource. Not only do we have experimental data on MPD, we also have protocols. And so we've got very detailed step-by-step protocols that explain how exactly these data sets were generated. And for a lot of examples, we actually have really nice pictures showing you what you should see when you perform this experiment in your own hands. Finally, I'll show you the importance of using genetically stabilized mice in your research. So here at the Jackson Laboratory, we have a patented genetic stability program in which we take frozen embryos and use them to refresh our foundation stocks about every 5 generations or so. These foundation stocks are then expanded and used for distribution to investigators worldwide. The reason we do this is to minimize the effects of genetic drift for some of our most popular strains. And so you can see that all of these strains here, in addition to some others that are not indicated here, these are all enrolled in our genetic stability program. So if you're using any of these mice for your studies, you can feel confident about the genetic quality of the mice that you purchase from us, and that the data that you generate from these mice will be relevant and reproducible, because the mouse that you're using now will essentially be the same mouse that you use 5, 10, or even 20 years from now. So I'd like to summarize what we've talked about today. First, I've shown you how important it is to choose the right inbred model for your infectious disease research and for what you plan on studying and modeling. Also, we've seen how inbred mice display the full spectrum of host susceptibilities to different infections. I've also shown you that you can use the mouse phenome database to help you with your strain selection. And finally, I've told you about our genetic stability program and how high health status in genetically stabilized mice increase the reproducibility of your results. With that, I'd like to thank you for joining us for our presentation today, and just wanted to remind you that not only do we sell mice, but we have a number of different mice-related services, and so you can take advantage of our expertise in performing some of these services, including highly customized and complex breeding projects, as well as compound efficiency studies. So if any of these things are of interest to you, please feel free to contact us or visit our website to find out more information. So I'd like to thank you again for joining us for our presentation today. And if you have any questions, you can always contact your regional representative by calling them or emailing them, or you can contact me or my colleagues in technical information for any mouse-related questions you may have, visiting our tech support portion of our webpage. Thank you so much.